really stopped to think about what you're made of? I mean, really, down to the last atom. Are we and all life on Earth just a random jumble of chemicals? Or is there a very specific, very special recipe? Well, today we're going to go on a kind of scientific detective story to unlock the chemical code of life. All right, let's get into it. So, what's our game plan? First, we'll look for clues in the basic elements that make up everything. Then, we'll do a bit of forensic analysis on living tissue. After that, we'll investigate the smallest building blocks and then the giants they create. We'll pay special attention to the master builders, the proteins, before we wrap it all up and see how this incredible chemical symphony comes together. All right, every good detective story needs a first clue, right? So here's where we're going to start. We're going to compare the list of chemical ingredients in living things, like us, with the ingredients in non-living things like, say, the Earth's crust. Are we really made of the same stuff as the ground we walk on? Let's find out. Okay, now this is where it gets really interesting. Just look at these numbers. Take carbon, for instance. In our bodies, a whopping 18.5% of our weight. But in the Earth's crust, it's practically nothing, 0.03%. That is a massive difference. And look at hydrogen, way more abundant in us. This isn't just a random mix. Something is clearly selecting these elements for life. This is our first major clue. So, what gives? This is the central question for our entire investigation. We're using the same basic elements, the same pantry of ingredients as the Earth itself, but the recipe for life is just completely different. Why? Why does life go out of its way to collect so much carbon and hydrogen? This is the mystery we have to solve. To crack this case, we've got to think like forensic chemists, right? We need to get a sample of living tissue, break it all the way down, and analyze exactly what's inside. So, it's time to get our hands dirty, scientifically speaking. So here's how it's done. You take some living tissue, let's say a piece of liver, and you grind it up with an acid. This acid basically just busts open all the cells and releases everything inside, making this thick soup, this slurry. Then, you just strain that slurry through a regular old cheesecloth, and boom, that one simple step separates everything into two neat piles for us to investigate. And here's what we get. The liquid that drips through, that's our acid-soluble pool. It's got all the tiny little bits and pieces, what scientists call the micromolecules. And the gunk left behind in the cheesecloth, that's the acid-insoluble fraction. And that's where the big stuff, the biomacromolecules, are hiding. Now we can really dig in and analyze each fraction. Let's start with that liquid fraction, the acid-soluble pool. Inside, we find thousands of different small organic compounds. Think of them as the fundamental nuts, bolts, and tiny little bricks that life uses to build everything else. So, our first suspect on the list, the amino acid. Now, they all share a common backbone, a basic structure that's the same in all of them. But here's the cool part. Each one has a unique little attachment called an R group. And that R group is what gives each amino acid its own special personality, its own function. And that R group really does make all the difference. Check it out. If that R group is just a simple hydrogen atom, bam, you've got glycine. Swap it out for a methyl group, and now it's alanine. Use a different one, a hydroxymethyl group, you get serine. It's amazing, right? Just by changing that one little piece, nature gets 20 different amino acids it can use to build all the proteins. Okay, next on our list, lipids. You probably know them better as fats and oils. Now, these guys are interesting because they generally don't like water. The simplest one is a fatty acid, which is really just a long chain of carbons. And when you stick 3 of these onto a little molecule called glycerol, well, you get a triglyceride. That's just the fancy science name for the fats and oils we use every day. And you can see the variety right here. On one hand, you've got something like palmitic acid. Then there's glycerol, that little backbone molecule we just talked about. But then look at this one, lecithin. See that phosphate group on it? That little addition makes it a phospholipid, and that makes it absolutely critical for building the membrane around every single one of your cells. And of course, we can't forget a few other key players in this group of small molecules. You've got your sugars, like glucose, that's life's main fuel source. You've got your nitrogenous bases, things like adenine and uracil. You probably recognize those from biology class. And then, when you put a base, a sugar, and a phosphate together, you get a nucleotide. And those are the building blocks for the big one, our DNA and RNA. Okay, so we've looked at the tiny bricks and bolts. Now, let's check out that gunk that got left behind in the cheesecloth. This is where the real giants of the cell live, the big, complex polymers that basically form the entire structure and machinery of life. And when I say giants, I really mean it. The difference in scale here is just huge. The micromolecules we just saw, they weigh less than 800 daltons. That's the unit scientists use. But these biomacromolecules, we're talking tens of thousands of daltons. They're massive. Oh, and a funny little side note, you know the lipids we just talked about? They're actually small, but they get caught in this fraction with the giants. That's just because when you grind everything up, they form these little bubbles that aren't soluble, so they get filtered out with all the big stuff. So if you were to take a cell and just break down what's inside, what would you find? Well, no surprise, it's mostly water, 70 to 90%. But look what's in second place. Proteins. They make up 10 to 15% of the cell. After water, they are the main organic ingredient. So yeah, they are clearly the star of the show here. We've got to take a closer look. Okay, let's put these proteins under the microscope. These are the master builders, the workhorses of the cell. They do, well, pretty much everything. But the real plot twist in our story isn't just what they do. It's how they're built, how they go from something so simple to something so incredibly complex. Everything begins at level 1, the primary structure, and it's super simple. It's just a straight chain of amino acids linked together end to end. That's it, kind of like beads on a string or letters in a really, really long word. But the specific order of those letters, that is everything. But you know, that chain doesn't just stay as a boring straight line. Nope. It immediately starts to twist and fold into these cool repeating patterns. One of the most common ones is this gorgeous shape, the alpha helix. It looks just like a spiral staircase. And that's level 2, the secondary structure. Then things get even more complex. The entire chain, with all its little spirals and folds, scrunches up on itself into this really specific 3D shape. The source material describes it as a hollow woolen ball, which is a great way to picture it. This is the tertiary structure. And let me be clear, without this exact 3D shape, the protein is useless. It has to fold correctly to work. And believe it or not, for some proteins, we're still not done. Some go to a 4th level, the quaternary structure. This is when you have multiple totally separate protein chains, each one already folded into its 3D shape, and they all come together to form one giant super complex. The classic example is hemoglobin, the protein that carries oxygen in your blood. It's actually 4 separate protein chains all working together as a team. And it's this incredible, multi-layered complexity that lets proteins do so many different jobs. I mean, look at this list. You've got collagen, which is basically the structural scaffolding holding you together. Then there's trypsin, an enzyme that just chomps up your food. Insulin, that's a hormone. You have antibodies, which are like your body's personal security force. And GLUT4, which is like a tiny doorway that lets sugar into your cells. It's just amazing what they can do. So there you have it. Our investigation is pretty much complete. We started with a simple question, what are we made of? And we found that while we use the same elements as the dirt under our feet, we use them in a totally different way. We saw how those elements build these tiny little micromolecules, and how those in turn assemble into the giant, incredible protein machines that run the whole show. Case closed. So yeah, we figured out the what. We know the ingredients. But you know, just like in any good detective series, solving one mystery just opens up an even bigger one. The next great mystery is how. How on earth does a tiny cell read its DNA blueprint to know exactly how to line up those amino acids in the perfect order to build every single one of these amazing protein machines? Now that, well, that is a story for another time.